and Andrew Shumway. This is the first of 5 in our series, UW Engage Science, and we're so pleased to be able to offer this platform to these students to present their important and engaging work. As we get underway, I'd like to acknowledge that our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continuing use of the natural resources of their ancestral homeland. Thank you for tuning in. Town Hall is proud to be a community-focused organization and a place where we can share and sustain ideas and creativity, even if it means we can't gather in person. I'd like to thank tonight's speakers for appearing tonight and helping make that possible. Tonight's broadcast is also streaming on our Facebook and YouTube pages. For viewers who want to watch this broadcast with closed captioning, we recommend viewing via our YouTube page. To enable real-time closed captioning, click the CC button in the bottom right corner of the video player. The video will also be available for rewatching immediately following tonight's broadcast on all of those platforms. Town Hall and the nonprofit community at large have been put under significant strain due to the recent wave of event cancellations. We hope you'll consider extending your generosity to help support us during this difficult time by making a donation or becoming a member. You can donate online or text Town Hall to 44321 to give. Town Hall's work is made possible through your support and the support of our sponsors. Our Arno Maltowski Science Series is supported by Microsoft, KUOW, Wyncote Foundation Northwest, and the taxpayers of Washington. Finally, Town Hall is a member-supported organization, so I'd like to thank all the members watching tonight. Tonight's program will include 3 separate presentations with a time for questions at the end. Each presentation is about 20 minutes long. You can submit questions tonight for our speakers at any time using the Ask a Question field at the bottom of your screen. If you can be sure when you put the question in to address the question to the presenter you intend it for, that would be greatly appreciated. We can't guarantee that we'll get to every question, but we will get to as many as possible. And now for our first presenter. Amelia J. DuVall spent the last several years getting pooped on while studying seabirds at Channel Islands National Park in Southern California. Now a graduate student at the School of Aquatic and Fishery Sciences at the University of Washington, her research on seabird ecology, conservation, and management across the Pacific Ocean from the Channel Islands to French Polynesia. Please join me in welcoming Amelia J. DuVall.
Hi, thanks. Thanks everyone for joining tonight, even though I do know that you have to be at home tonight anyway. But like Candace said, my name is Amelia DuVall, and I'm a graduate student in the Quantitative Conservation Lab at the University of Washington. And this photo of me was taken in Channel Islands National Park in Southern California, where I spent several years doing habitat restoration and seabird research. And I titled this talk, She Sees Seabirds by the Seashore, partially as a joke, because I knew my tongue would get tripped up on the words, but it also does a surprisingly good job of describing my research. I spent a lot of time looking at seabirds through binoculars on islands and also at sea from boats and planes. And so I became a seabird biologist for the obvious reasons. It's a really glamorous job. That is poop on my face. And I think the most telling part is that I would stop to take a photo before wiping it off. And this is definitely not the first time this happened either, or the last. The true reason why I became a seabird biologist has more to do with this. This little fluff ball is a 1-day-old Scripsis merlet chick. And any brand new chick is cute, but this one is extra special because you rarely get to see these chicks at all. Just one day after they hatch, they follow the sound of their parents' calls, leaving their nest in the middle of the night and tumbling down cliff sides until they land in the ocean, where they reunite with their family. And they will then spend the rest of their life out at sea until they're ready to come back to land to lay eggs and raise their own chicks. But to be honest, I also became a seabird biologist because of moments like this. The opportunity to be so close to these creatures that you usually only see from a distance as they are flying over you, and hopefully not pooping on your face. But I did not grow up loving seabirds or even birds. The place that I first fell in love with seabirds is Channel Islands National Park, which I began visiting when I was 18 years old. And at first I came to these islands for my own benefit, like to scuba dive or go backpacking. But over time, I realized how important these islands are as a refuge for seabirds and many other plants or animals that have very little habitat left over in Southern California, where so much land has been developed and paved over. And to orient you a little bit, there are a group of 8 islands off the coast of Southern California, known as the Channel Islands. And 5 of these that you see circled here are a part of Channel Islands National Park. And when I say a lot of seabirds nest at Channel Islands National Park, I mean a lot. There's somewhere between 50 to 100,000. It's actually really hard to get an exact number on how many, but we do know that the park provides nesting habitat for 99% of the seabirds in Southern California. If it weren't for these islands, these birds would not have anywhere else to nest and raise their chicks. And there are at least 13 different seabirds that nest out on these islands. Some of these you might already know, like the pelican, but this mix of seabird species does not exist anywhere else in the world. But tonight I'm only going to talk about 3 of these seabirds. I'm going to talk about the California brown pelican, ashy storm petrel, and Scripps' merlin. You might recognize that chick from earlier. So these seabirds are particularly important to the Channel Islands because of how much their populations rely on the Channel Islands. The only place where the California brown pelican nests in the United States is the Channel Islands, and only on 2 islands out there. About half of the ashy storm petrel population nests on the Channel Islands, and the only place where the Scripps' merlin nests in the United States is also on the Channel Islands. And one of these islands probably has the highest number of Scripps' merlin nests in the world. And so Channel Islands, they're a really important place to conserve these seabirds. And national parks are responsible for managing and conserving the plants and animals within them. But there's only so much that the park can control. The park can limit development and research and visitors, but it can't keep things out like climate change or plastic pollution. Some seabird species nest in caves where they're sheltered from wind, rain, and sun. These caves are closed to the public because it's very easy to crush a nest or spook a seabird as you walk in without knowing it was there. And these signs can keep people out for the most part, but every year we find more and more plastic and trash piling up that has washed up from who knows where. And one of the reasons seabirds are so unique is because they live their lives in 3 different worlds. They lay their eggs on land, they fly in the sky, and they feed in the ocean. But this means, however, that they're vulnerable to threats in each of these places. On land, many seabirds nest directly on the ground, where they're vulnerable to non-native predators like rats, mice, and cats that have been introduced by people to parts of the world where they never existed before. These non-native predators eat seabird eggs and chicks, and even adults sometimes. And seabirds are also losing precious nesting habitat on land due to sea level rise and development. And in the sky, seabirds can collide with offshore wind energy turbines that they don't see, and they also suffer from light pollution. Some seabirds are nocturnal and they fly around at night, but their vision is messed up when they encounter bright lights on boats, and they can crash right into them and become disoriented. And in the ocean, seabirds are vulnerable to changing ocean temperatures, which can affect which fish are around and how many. If ocean temperatures change too much, it can be more difficult for them to find food to eat. I'm going to talk about 2 of these threats on land, non-native mammals and sea level rise, and what we can do about them later on in this talk. Due to these threats, seabirds are the most endangered group of birds in the world. About 70% of seabird populations have been lost between 1950 and 2010, and that's 230 million seabirds. And this statistic might make your heart drop, like it does for me. For me, I feel a basic responsibility to protect these plants and animals in the world, especially since so many of them are suffering from our own doing. But there's another reason why we should be paying attention to seabirds. We stand to benefit from watching them. Seabirds are known as ecosystem sentinels, which means that they can tell us things about the environment that would otherwise be more difficult for us to see ourselves. Things like pollution, changes in ocean health, and the status of fish stocks. These are all things that affect seabirds, but they also affect us. I think it's helpful to think about this as a car's dashboard. A car is this complex piece of machinery. Most of us don't really understand how they work, and it's hard to take a good look on their insides. And to me, the ocean is similar. In our cars, we don't have to constantly check our fuel or coolant because in most cases, the dashboard will light up and tell us if something is wrong or needs a closer look. And seabirds can do the same thing for us for our oceans. Seabirds are sensitive to changes in their environment, and if we see a change in seabird health, that's an indication of a bigger problem in our oceans. And you might be thinking, isn't this just like a canary in a coal mine? But seabirds are so much better than a canary in a coal mine because a dead canary can only tell you so much. By using seabirds as our indicator lights, we can get an idea of what the problem is, where it is, and how bad it is, just like your car can tell you if the issue is with your engine or your tires or your brakes. And let me give you an example. So this is a pelican egg. I've got really small hands, but you can still tell that this is a pretty big and solid egg. But for some reason, back in the 1960s, hundreds of pelicans across the Channel Islands started crushing their eggs when they tried to sit on them. And it was because they had been eating fish that was contaminated with DDT, a pesticide that was really popular back then. And it turns out a company was dumping wastewater with DDT into the ocean, and the pelicans were eating fish contaminated with the pesticide. This caused pelicans to lay super thin eggs and all kinds of other problems. People were eating fish from the ocean too, and DDT in large amounts can be harmful to humans. Now, you can't see DDT in the ocean, but you can see crushed eggs and total reproductive failure for pelicans. A seabird watching the pelicans used this information to make the case against the company that had dumped the DDT into the ocean. And as a result, a settlement went towards cleaning up the DDT pollution and helping the animals that had been harmed by it. But more recently, you might remember the blob of warm water that sat in the Pacific Ocean near our coastline a few years ago. Massive seabird die-offs was one of the first indications we saw that there was a problem in our ocean. These seabirds warned us that there was a problem, and later on, we saw all these cascading effects in our commercial fisheries, like the shutdown of crabbing and clamming industries and lower returns of salmon. If we watch seabirds, we can get an idea of when and where these events are going to occur and how we can better prepare for them. So how do we keep checking our ocean's health by watching seabirds? This is where Seabird Monitoring comes in. Each seabird species nests in different places and has different behaviors, so we do different things in order to monitor them. And part of my graduate research is working with Channel Islands National Park to help them figure out how seabirds are doing and what they can do to help them. So on the easier side of the monitoring spectrum, the California brown pelican builds a large nest right on the ground. It's a big bird, so it's a big nest that's easy to spot. But thousands of pelicans will build their nest on one small island, and they blend in really, really well, which makes it really hard to count them and see them all. You might see a pelican here or two, but if you look closer, there's at least 13 by my count. And even worse, sometimes we are trying to count all of the pelicans from a boat. And the rocking back and forth of the boat as you stare through your binoculars at the land makes you want to vomit. And the sun is usually shining right in your face too, which does not help. Oftentimes you end up losing track somewhere where you're at 32 or 235 or somewhere, and you have to start all over again. But sometimes the best way to get an idea of how many pelicans there are is to take photos from above. And I probably don't need to tell you that this is way more fun than counting from a boat. So then we can take these photos and upload them to a computer program that allows us to count up the number of nests and chicks that there are in each photo. And we can see how many pelicans showed up to nest that year and how many chicks they raised and compare this to other years to get an idea of how they're doing. Ashy storm petrels, on the other hand, are a smaller bird that is much harder to track down. They nest in small crevices that are difficult to get to, like rocky cliff faces, offshore rocks, and in sea caves. And they go to and from their nests at night when they are less likely to be spotted by predators, as well as pesky researchers trying to study them. So we can take boats out to some of these places to take a look at their nests, but even then, we have to do all sorts of crazy maneuvers to try and take a look at their nest and see if anyone's there. We get into lots of super flattering positions like this one too. But despite our best efforts, we still can't reach all the nests, So in order to get an idea of how many individuals there are, we have to get more creative. As I mentioned earlier, the ashy storm petrel is nocturnal, and it only comes out at night from its nest. So to get an idea of how many birds are nesting in an area, we string up this fine net called a mist net. We open it up in the middle of the night, and we use the sound that ashy storm petrels make to bring them into the net. This net doesn't hurt them, and we release them back into the wild as soon as we're done. But first, we have to carefully remove them from the net. These are small birds, but they can put up a surprisingly good fight, and they have a tendency to vomit up this orange, slippery, foul-smelling seafood goop that only makes the net more slippery and the task more challenging. But once we finally have them in hand, we can take measurements that tell us how healthy they are and place a band on their leg that lets us identify individuals over time if we're able to capture them again. And so this is what Seabird monitoring data looks like on the back end. It can be kind of messy. I'm taking this information that we collected from monitoring, and I'm looking at the results over the years to see how numbers of seabirds are changing over time. These changes might tell us if there are any new seabird indicators of ocean health that maybe we should pay attention to. I'm also looking at the current impact of the threats I mentioned earlier and seeing how these threats are affecting seabirds now as well as into the future. And this will help the park figure out which seabirds are having trouble and why, and what they can do to help them. Because there's a lot that we can still do to help seabirds. And this brings us to the last bird, the Scripps merlin. So this bird is also nocturnal like the ashy storm petrel, and it nests in rocky crevices too, like you can kind of see in this photo. And this species is pretty rare. They only nest on 12 islands off the coast of Southern California and Mexico. And here's a photo of some Scripps merlin eggs. I should tell you that these eggs are numbered for monitoring purposes. They're cool birds, but they do not lay magically numbered eggs. The reason I'm showing you these eggs is that so you can see how they're right out in the open and how the adults are not always sitting on top of them and protecting them. And this means that it's really easy for this to happen. The Scripps merlin was almost wiped off of one of the islands by non-native rats that ate their eggs and chicks. These rats were likely brought to the island by mistake during a shipwreck in the 1800s. The seabirds here had never experienced rats before and didn't have the right defenses to protect themselves in their nests. But in the early 2000s, the National Park Service removed rats from the island, and since then, the Scripps merlin breeding population has more than quadrupled. And now, instead of seeing rats on camera, we see this happy ending, which is a little merlet chick making his way out to sea with his parent in the middle of the night. Another threat that I mentioned earlier is sea level rise. And a way we're working to help seabirds with sea level rise is by partnering with artists and others to create artificial habitat for seabirds. And these structures provide a safe space for seabirds to nest in while they're losing natural habitat due to development, drought, and sea level rise. The structures seen here are known as bread loaves due to their shape, and they're handmade by students at the California College of the Arts. And each bread loaf contains 3 small chambers for seabirds to nest in. First, we'll usually see an adult in the artificial habitat like this ashy storm petrel, which is a good sign that someone has decided to use it as a nest. And then, if we're lucky, about a month or so later, we find little chicks in the nest as well. And each year we find more and more chicks in these structures, which means that seabirds are finding them and successfully using them to raise their babies. The lessons that we learn aren't just staying in the Channel Islands either. I've been lucky enough to start studying seabirds across the Pacific Ocean in French Polynesia on a small coral atoll called Tetiaroa. And Tetiaroa is located way out in the South Pacific, about 30 miles north of Tahiti. And 12 species of tropical seabirds nest here year-round. And I recently began working with researchers at the University of Washington to study seabird health here. It turns out that rats were also introduced to this place by people, just like in the Channel Islands. And just like in the Channel Islands, most of the seabird species here don't actually build a nest and instead lay their eggs and raise their chicks right on the ground. So you can see why rats are really doing a number on these vulnerable seabirds. But the great news is that rats are going to be removed from Tetiro next year. And they're using the same methods that were first used in the Channel Islands to remove rats there. And as part of my work here, I'm studying how seabirds change after an eradication to help support other future rat removal projects around the world to benefit seabirds. So earlier I made this comparison of seabirds to a car dashboard. And I think that the metaphor helps to get this idea across, but there's actually one major problem with it. And that is when our cars break down or the check engine light won't go off, it's often easier to just fold your hand and go out and buy a new car. And I used to drive an old Volvo, and I'm very familiar with the check engine light. So eventually I had to fold my hand and I went out and got a new car. That was actually a terrible idea since I'm now a poor graduate student. But the reality is we can't go out and buy a new ocean if we don't take care of the one that we have. And for me, I would prefer to pay attention to the dashboard and keep our ocean healthy and running instead. So thanks so much for your time. I want to thank all the other seabird researchers out there and the photographers that let me use their photos. And I hope you enjoyed this story. And yeah, thanks. I'll turn it over to the next presenter now.
All right. Thanks so much, Amelia. I guess where the people go, the rats go. So it's good to know that. And I'm so happy you're working on that. Our next speaker is Anne Polyakoff. Anne is a graduate student in the Quantitative Ecology and Resource Management program at the University of Washington, and she explores how plants communicate using vast underground fungal networks. Anne studies the costs and benefits of participating in these networks and what we can learn from plants by listening to what they are saying in the Wood Wide Web. So please join me in welcoming Anne Polyakoff.
Thank you, Candace. Imagine you are standing in a beautiful forest. Trees surround you. You might hear birds singing or the quiet rustle of the wind through the branches. It is probably a quiet place, and it doesn't seem like there is much going on around you. But a forest is much more than what you can see or hear, because the plants and animals of the forest have evolved to communicate with each other in a different way than we do. The forest is a place of intense activity, movements and messages that are totally invisible to our eyes. And just because we can't see them doesn't mean they're not there. If you look beneath your feet in the soil, there's a whole world pulsing with life, a marketplace where food is traded, and a network through which messages are sent throughout the entire forest. And you don't have to dig that far down to find this world. In fact, if you just scoop away some soil, you will likely come across a thick mat of tiny white strands. Sounds pretty weird, right? Well, these white strands are fungi, and you might better know fungi as mushrooms, chanterelles, morels, portobellas. Mushrooms are the fruiting structures of fungi, like apples are for apple trees. And even though these fungi look tiny and insignificant, they play a huge role in shaping forests across the entire planet. In fact, these tiny fungi helped plants colonize land about 450 million years ago, which is what led to us being here today. But what makes these tiny creatures so important? Well, a specific kind of fungi has learned to form a unique partnership with plants, and this fungi is known as a mycorrhizal fungi. Now, the word mycorrhizal stems from the Greek word mycos, meaning fungi, and rhizal, meaning root. So, fungus root. And this description is accurate, because if we zoom into these roots, we can see that the plant and the fungi don't just intertwine around each other, but literally fuse together on the most fundamental building block of both plants and fungi, the cellular level, to form this intimate and fascinating partnership. The plant cell is the bubble-like structure in the image to the right, and the fungus is the green thread weaving through the plant cells. Now, these cells are so small that just the tip of a ballpoint pen would hold 100 of these plant cells. Now, the reason why plants and fungi form such an intimate relationship is because each has something that the other does not. Plants use sunlight to make sugars in the form of carbon, which fungi need to grow and survive but cannot make themselves. Fungi, on the other hand, are much more effective at acquiring nutrients, which you can think of as vitamins or supplements. These fungi can spread through the soil quickly when searching for nutrients and transport them over long distances through their thin strands. Plant roots need these nutrients to grow, but can't extend their roots like fungi can. Thus, plants give fungi sugar in return for nutrients, and both rely on the other to grow and survive. But each fungi doesn't just partner with one plant, and each plant doesn't just partner with one fungi. In fact, one fungal individual can partner with dozens of plants, and one plant can partner with dozens of fungi. Which, if you think about a good business partnership, makes sense. You want to diversify your portfolio so that if one business partner pulls out or one business venture fails, you still have some assets. The same thing happens with plants and fungi. Scientists have shown that when a plant gives the fungi carbon and the fungi doesn't give anything in return, the plant will cut off the fungi from its carbon supply and network. The same has been shown for fungi. So both partners have a say in the terms of this relationship. But what's even more astounding is that these fungi link the roots of different plants together and form what are known as mycorrhizal networks. These networks play a huge role in plant communication and are sometimes known as the Wood Wide Web. By connecting plants together underground, these fungal networks serve as the information and resource highways that plants use to communicate with each other. Yes, you heard that right. Plants do talk with one another, but in a very different way than we do. While we use sounds to communicate, plants use chemicals to talk with one another. And unlike us, plants have had millions of years of evolution to refine their chemical language, and they are masters of chemistry. By sending different sequences or cocktails of biochemical signals within these networks, plants can tell each other when they're hungry and need food, they can warn each other when danger is close by, and they can even recognize and nurture family members. Now, I study how plants warn each other of incoming danger, which is known as defense signaling. Danger for plants primarily implies insects, which come to eat leaves and other plant parts and can cause major damage and even death. When a plant is injured by an insect, it quickly produces defense chemicals, illustrated by the green ovals here, that it then uses to kill attacking insects and repel incoming insects. But the injured plant also sends some kind of defense signals through these mycorrhizal networks to healthy plant neighbors. The healthy neighbor plant can then increase its own defense chemicals before it's attacked, thus receiving this early warning signal. But the strange thing is that we still don't know exactly what kind of signaling occurs through these networks. The actual signal has never been measured. All studies use a substitute for this defense signaling by measuring the amount of defense chemicals produced in the injured plant and in healthy plant neighbors. And they find that without the mycorrhizal fungus, the neighboring healthy plant doesn't get this early warning signal from the injured plant. Now, there are a few theories or ideas for what is actually occurring below ground. The first and most widely known theory is called active signaling. In this theory, the injured plant actively sends chemicals to healthy neighbors through the fungi. The injured plant thus communicates or talks directly with its neighbors. In the passive signaling theory, defense chemicals from the injured plant leak down through the roots and move some way through the fungi, which a neighbor plant then senses and responds to by producing its own defenses. Here, the signal is not actively communicated to neighbors, but neighbors listen in on each other. Another theory posits that perhaps the fungi really control the transfer of signals between plants. You see, when the plant is attacked by insects, it needs a lot of energy in the form of carbon to create defense chemicals. This means the plant has less carbon to give to the fungi, which needs this carbon to survive. The fungi could pick up on this rapid decrease in carbon and signal a defense alarm to a neighbor plant. The neighbor plant can then increase its own defenses, making it more likely that it will fight off the insects and ensure long-term carbon supply to the fungi. So here, the fungi is really looking out for itself. Now, I'm especially interested in understanding how trees and forests signal to each other about incoming danger. And I think this is especially important because in recent decades, forests have been experiencing unprecedented insect attacks on a massive scale. And warning each other of danger could be an important way in which forests respond to these attacks. Now, when I first got started with this research, I was really excited because there were tons of news articles about how trees send defense signals to other trees and forests, so I thought I had a lot to work from. But as I dug more into the research, I found something that was really surprising. Almost all defense signaling studies have been done only on small herbaceous plant species, such as tomato and bean plants. I wasn't able to find any studies that demonstrated defense signaling in trees. Well, I thought, okay, that's okay. I mean, tomato plants, trees, tomato, same thing, right? But the more I thought about it, the more I realized that there are huge differences between trees, organisms with a large woody stem, and small, fragile tomato plants. While herbaceous plants such as tomatoes and beans are small and typically only live a few years, trees grow to be some of the largest organisms on Earth and can live for hundreds of years. These and other differences between the two plant types suggest that trees could respond very differently to insect attacks. Now, after poring over many studies, I did finally find a study that looked at defense signaling within trees. The study found that when a Douglas fir seedling or baby tree was injured by an insect, it warned a neighboring pine seedling through the fungal network. Okay, I thought, this is great. Defense signaling has been shown in trees. Now I can really get to work. But then I did some more research, and that's where I ran into the same problem as before. You see, it turns out that small tree seedlings, or small herbaceous plants without a woody stem, such as tomato or bean plants, respond to insect attack differently than large adult trees. When a small plant is attacked by insects, it produces defense chemicals throughout the entire plant, leaves and stem. But when a large adult tree is attacked by insects, the tree only produces defense chemicals in the area surrounding the insect attack. A tree's defense response is thus localized, meaning defense chemicals are produced only at the attack site, but not throughout the entire tree. And this is because it would take the tree a huge amount of energy to send defense chemicals throughout its entire body. So if the defense response in a tree is so localized, how would a tree get a signal down to its roots and across the fungal network to another tree to communicate with it? At this point in my journey, I was feeling pretty defeated. Does defense signaling in trees even occur at all? And that's when it hit me. You see, a tree produces its defense chemicals in the phloem. And just like blood vessels that transport materials and blood throughout the human body, the phloem is the main tubular structure in trees that transports food and other signals from leaves to roots of that tree. We think that it is very likely the phloem transport stream picks up defense chemicals and carries them down to the roots through the fungal networks and to neighboring trees. We're really excited to test this idea at the Charles Pack Experimental Forest in Washington this summer. An experimental forest is an area of land set aside as a living laboratory where scientists conduct experiments to help better understand and ultimately protect our forests. We will be setting up a study to measure the defense response of trees injured by insects and neighboring healthy trees to better understand how trees use these mycorrhizal networks for defense signaling. But we're not just interested in the defense messaging that occurs within these networks. We're also interested in how plants use these networks to send food to each other, especially when some plants have a surplus of food and other plants are in need of food. To study this, we decided to look at a system where there is an influx of nutrients that occurs every single year. And that system is a salmon stream in a forest. Every year, hundreds of salmon return from the ocean to the streams where they were born. When the salmon get back home from the sea, they mate with other salmon and then die. As the dead salmon break down in the soil, they release a huge amount of nutrients, valuable nutrients on the banks of the streams. These nutrients can be traced through the soil, the fungi, and the trees. And there's evidence to suggest that plants closer to the stream are uptaking these salmon nutrients and sending them through the fungal networks to trees further away. We're studying this at a really cool stream located in a beautiful boreal forest in Alaska, where an exciting study has been taking place for some time. For the past 20 years, scientists have taken all salmon that have returned to the stream and thrown them onto one bank of the stream after their death. This creates a huge amount of salmon nutrients available to plants on one side of the stream, but not the other. We want to test if the trees on the salmon bank are redistributing these salmon nutrients to trees further away from the bank. And we're really excited because we're finding these salmon nutrients in trees far away from the bank where the salmon were thrown, which suggests that these nutrients are being redistributed into the forest to trees away from the stream edge. We believe that mycorrhizal networks might be helping the trees in doing so. This really shows that this is a highly interlinked system. Fish, forest, fungi come together to provide. I think studying these plant fungal partnerships and networks can really remind us that we are all dependent on each other and that we are more interconnected than it seems at first glance. We still don't know a lot about what the plants and fungi are saying to each other, but we do know that plants and fungi have been talking for millions of years longer than us, and they've had a lot of time to come up with clever solutions to problems along the way. Plants and fungi have already helped us find life-saving antibiotics such as penicillin. What other cures could they provide? Fungi also store vast amounts of carbon in the soil. Could they be a solution to our carbon emissions problem? Who knows what else their conversations might tell us? Perhaps if we listen closely, we'll find out more about our own roots. So next time you're outside and walk by a plant, whether it's a flower or a pine tree, you may wonder what that plant is talking about right underneath your feet. Thank you.
Thanks so much, Anne. It's so exciting to think of plants having human characteristics like communication. So thank you so much. And animal communication tactics. I just want to remind everyone that we are going to be going to questions at the end of this next presentation. So if you do have a question, be sure to put it in the Ask a Question field at the bottom of your screen, and just be sure to address it to the presenter you intend it for. So our final presentation comes from Andrew Shumway. Andrew is an astrobiologist and planetary scientist who is fascinated by the role of water on Mars. His experiments recreate the cold, dry conditions of Mars' surface to study how water forms in extreme environments. Andrew is originally from Atlanta, Georgia, and is currently working toward a PhD in Earth and Space Sciences and Astrobiology. Please join me in welcoming Andrew Shumway.
Hi, everyone. Thanks for that introduction, Candace. All right. I want you all to imagine for a moment that I am the CEO of a space travel company, and I've spent billions of dollars building a rocket to safely take humans to Mars for the very first time. And to thank you all for tuning into my talk today, I want to offer you all a ticket to join me for a few weeks on the Red Planet. So take a moment to mull that over. Would you want to visit Mars? Maybe not everyone's convinced, and for good reasons. Mars is not an ideal spot for a vacation. To understand some of the challenges of living on Mars, let me take you down to the planet's surface. Welcome to Mars, where it's the hottest day of summer and the temperature outside is a balmy 32 degrees Fahrenheit. For the rest of the year, the temperatures are colder than Antarctica. There's no oxygen in the air, and in fact, there's very little air at all. Because there's no oxygen, there's no ozone layer to protect us from the sun's deadly radiation. And a few minutes outside would give you severe sunburns. Longer exposures would definitely lead to cancer. But the good news is that we have solutions to overcome most of these challenges. That, for example, we can build structures or wear spacesuits that block the sun's rays, keep us warm, and give us enough air to breathe. But there's one major problem that we just haven't solved yet. On a planet that's drier than the Sahara Desert, where are we going to find the most important ingredient for life? Water. So unfortunately, I'm not actually a billionaire. I'm a grad student and I don't have a rocket ready to take anyone to Mars. But whether or not you personally would want to go to Mars, one day soon, humans are going. Our children or grandchildren could be the first humans to ever set foot on Mars. And when they get there, they're going to need water to drink, to shower, to water crops and grow food. If we were just visiting Mars, we could bring enough water with us to last the entire trip. But what about the long term? It would be impossible to bring a lifetime supply of water with you from Earth. So instead, we have to find water there. Today, Mars is a desert planet, but it hasn't always been that way. There's lots of evidence that Mars once had rivers and lakes just like we have here on Earth. We can see some evidence of this water by looking at satellite photos of Earth and Mars and comparing them to each other. So here I have two similar satellite photos in black and white. On the left, we have an image of Earth, and on the right, one of Mars. Both of these photos show a delta, which is a specific type of erosion pattern that's caused when a river reaches a standing body of water, such as an ocean or a lake, and branches out. Just by comparing these photos, we can see that a long time ago, an ancient river here on Mars flowed into a lake and made this delta feature. But today, both the river and the lake on Mars are dried up. There are also extensive river networks on Mars, just like here on Earth. Imagine how much water there must be, or must have been on Mars in order to create these elaborate valley networks with smaller streams flowing together and merging to form massive rushing rivers. Apart from the dried riverbeds and deltas and lakes, there's an even bigger sign that water once flowed on Mars. The color. The red planet gets its nickname because of all the rusted minerals on the surface. Just like how a bike might rust when you leave it out in the rain for too long, the iron minerals on Mars were exposed to water for so long that the entire surface of the planet rusted over. Just take a moment to imagine how much water it must have taken to rust the entire planet. From all this evidence, it's clear that Mars once had flowing water, just like Earth, but today it's all dried up. So there won't be any water for our future astronaut grandkids to use. So why am I telling you all this? Well, it's useful to track down what happened to all the water. And in order to answer that, we're gonna have to zoom out a bit. So here I have photos of the Earth and Mars, roughly to scale. Earth is full of all kinds of water. We have frozen ice at the poles, we have oceans all over the surface, and water vapor up in the air forming clouds. But on Mars, it's a different story. There's no liquid water. There's some ice frozen up at the North Pole, and a very, very small amount of water in the air. This is because Mars is further away from the sun, which is our planet's source of heat. Even on a warm summer day at the equator, the temperature on Mars hardly makes it above freezing. So whatever water there is on Mars, we should expect to be frozen solid year-round. Except I'm here to tell you that that's not the case. And even at these extremely cold temperatures, we might be able to find liquid water on the surface of Mars. To understand how this is possible, let's think about winters here in Washington. Up in the cold mountain passes, when wet roads freeze solid, how do we melt the ice and make the roads safe to drive again? We salt them. This is because adding salt to ice causes the ice to melt and form salt water. Well, okay, we do our best in Seattle. But the idea here is that salt water is much harder to freeze than fresh water. And this is the same reason why lakes up in the Cascades might freeze in the winter, but you'll never see Puget Sound freeze over. Another way that salts form water is by pulling it straight out of the air. This is a video of some balls of salt that are about ½ an inch across. If you've ever left table salt out on a humid day, you might notice that it gets clumpy and sticky after a while. What's happening is that the salt is pulling water vapor straight out of the air. The salt absorbs this water vapor, which starts it to dissolve it into the salt water. And under the right conditions, the salt can keep absorbing more and more water until it just dissolves completely, leaving behind very salty droplets of water, like we see here. And this is happening on Mars today. It's one of the ways that water can form there. But it's a little bit different on Mars because we don't have to go out and spread salt in order to turn ice into water. It turns out that there's already salt there, naturally occurring in the soil, that pulls water vapor out of the air to form salty water. Now, on Mars, there are more types of salt than just your average table salt. Many of these salts exist on Earth too, and you might be familiar with some of them. For example, there's magnesium sulfate, which is more commonly known as bath salts, nitrates, which we use in our fertilizers, and perchlorates, which we use in roadside flares and fireworks. All of these salts have been detected to naturally occur on Mars. So we know that salts can form liquid water under extreme conditions, and we saw that the surface of Mars is covered in salt. But what we don't understand is how much water is hiding in the dirt there. So to answer this question, I went to the lab to try to recreate Mars-like conditions and study how that water behaves. But in order to recreate a Mars-like environment, I need two ingredients: salt and soil. The salt was easy to find. Like I said, most salts on Mars also exist on Earth. I chose a salt called perchlorates, like the ones we use in roadside flares, because it is the most abundant salt in Mars's dirt. But finding the soil is a little bit more challenging. We've never actually returned any mission from Mars, so I don't have any real Mars dirt to work with. So I have to go with the next best thing. We do know what the dirt on Mars is made out of because past rovers have dug it up and analyzed it and studied what's in there. In order to recreate similar dirt as to what we would find on Mars, NASA scientists searched all over Earth for the closest matching rocks. And their search brought them to the Mojave Desert in the southwestern United States, which, if you squint your eyes and ignore the trees and the clouds and the blue skies, it kind of does look like Mars, right? But more than just looking like Mars, the rocks here are as close of a match to Mars as we can get, down to like the chemical level. So there's a company that takes these rocks, grinds them up into sand, packages it up, and sells it. It's what NASA uses to test their rovers. It's what I use in my experiments. And if I ever have kids, they're going to have the coolest sandbox on the block. So essentially, through these experiments, what I'm doing is recreating Mars in a jar, which you can see on the left here, a photo from one of my actual experiments. Inside of this jar, I can adjust the conditions, changing salt content, the amount of water vapor in the air, and the temperature to study what happens under different conditions. And by doing this, I can paint a full picture of what's going on Mars, here on Earth. So let me illustrate what happens when I adjust these knobs. If I add a lot of salt to the soil, for example, what I see is that a lot of water vapor gets pulled out of the air and absorbed by these salts, which then dissolve into salty water. Similarly, when I crank up the humidity and put in a lot more water vapor into the air, even more water gets pulled out of the air. So the salts absorb even more. Next, what I want to know is whether these salty soils are wet or if they're frozen and icy, because Mars is really cold, right? So what I do is adjust the temperature. I can turn down the temperature to hundreds of degrees Fahrenheit below zero, just to see how cold can it get before these briny waters freeze over. Through these experiments, I found that there could be a lot of water hidden on Mars. It's just been hiding in the soil. The salts we find on Mars are very, very good at absorbing water from the air and keeping it liquid down to very low temperatures. So you might be thinking, great, all right, there's salty, wet dirt on Mars, but what does this mean for our astronaut grandchildren? They're not going to eat a bunch of salty, wet dirt. What they need is fresh water. So in order for this to be of any use, they'll have to separate the water out from the salt in the dirt. One way to do this is through heating. Heating the soil will cause the water to evaporate, leaving behind the salt in the dirt. After that, you can collect the evaporated water into a separate container and cool it down so that it condenses back into pure water. And voila, that's it. That's one solution to the problem of how to get water for our future astronauts. But it's not the end of the story. There's another problem here. Humans aren't the only creatures that use water to survive. What if we get to Mars and find out that there are already microbes living in the dirt there, surviving off of this salty water? This is a really serious concern for NASA. If there is life on Mars, we have a responsibility to not interfere with it. And if there are Earth germs on the Curiosity rover, for example, it could act like an invasive species and kill off native Martian life. NASA takes this so seriously that they actively steer the Curiosity rover to avoid any areas on Mars where they even think there might be water. So part of my work is to ask, if this water exists, which I think I've shown it does, how salty can it be before it's no longer useful for life? Would life be able to survive in these salty, wet dirts, or is it too salty? So here I have a scale to measure how salty a solution is. How much water is available in a particular solution. So down here at 0%, we have pure salt, no water at all. And up at 100%, you can think of this as like pure mountain, spring, the freshest water you can get. So let's think about where the Dead Sea would fall on this. So the Dead Sea is one of the saltiest bodies of water on Earth, and it gets its name because it's so salty that no plants or animals can live there. There are still little microorganisms like fungi and bacteria that can live in the water, but for the most part, it's completely devoid of life. So where do you think that the Dead Sea would fall on this scale? It falls at about 98%, so a lot closer to fresh water than I originally thought. Okay, so let's think about what is the absolute lowest limit where you can have life exist in a salty pool of water. They've done experiments, biologists, with all different types of organisms, and they found that at about 60% on this scale is too salty. Life as we know it, nothing can grow below 60%. So in my experiments, in my little Mars in a jar, what I've tried to do is fit that water onto this scale. And I found that the salty water on Mars entirely falls below this 60% threshold where we know no more life can survive. What this means is that it really doesn't look good for our Martian microbe friend. So just to leave you with some concluding thoughts, there is a lot of water on Mars, but it's just been hidden in the soil. And future explorers might be able to use this water to survive in the cold, dry environment on Mars. Lastly, I just wanted to thank you all for attending this virtual talk and thank Town Hall for hosting us. We're really excited to have a space to share our science with you all.
Great, thanks so much, Andrew. So now we're going to move into questions. If you give us just a moment, we are going to invite our other two presenters back on. If you have any questions, I'm going to go in order and kind of group them. So we're going to start with Amelia.
Okay, can you hear me?
Yes.
Okay, cool.
Great. So let's see what we have here. So this question comes from Jenna, and it is for Amelia. What sorts of methods do people use to remove rats from the islands?
Yeah, so one of the methods that's become is more used right now is actually the use of toxic bait. And the other methods that people have tried, like trapping, it won't work because rats can go everywhere. And if you leave just one rat, it could be a pregnant female, and you are back in the exact same place where you started, except you've just spent a lot of really, really rare conservation dollars. And it's really important to use these methods that we know work because, like I said, these are really expensive projects and you don't want to fail. But I'll admit, like, it's a really hard conservation decision to make to kill one species to benefit another. But the reality too is, like, we're not just talking about hurting seabirds. Rats also hurt native plants in areas because they can eat the seeds of native plants and prevent them from recruiting. They'll eat native reptiles, and they can also harm coral reefs because seabirds, the nutrients from their guanos can help the productivity of coral reefs. And in some places too, rats are hurting local livelihoods because they're eating food crops that people depend on. So, like I said, it's a really hard thing to talk about and it's a hard thing to defend, but I think it's a human-induced problem, and it's up to humans to fix it. And one last thought on that too, like these other threats I talked about, like climate change, I mean, there's really not that much we can do to help rare birds or plants and animals with something like climate change. It's like such a huge problem. But we can do things like these eradication projects to help these plants and animals and put them in a better position to buttress them against some of these other threats that are a lot harder for us to deal with. Yeah, so that was a little bit of a tangent, but hopefully that answers your question.
Yeah. This next one is from me, actually. So you are studying birds, seabirds that are living on islands. Are there other extensive studies just as yours, say for seabirds that are not on an island, or why is it more effective to study a seabird from an island?
Yeah, I mean, so actually, islands are one of the few places where seabirds historically have been protected. So on a lot of islands, there aren't any native mammals. There are these isolated places that seabirds could fly to, but over time, no other mammals could get to them unless they happened to float over or something like that. So even before people started bringing mammals over to islands, islands have always been an important place for seabirds. And in terms of conservation actions too, like I was saying, we're at a spot where we have to prioritize because there's so many environmental problems. You can't fix them all. And islands, I think, are a really great place to work because you can kind of control it to some degree. If you try to do like an eradication on the mainland, it's a lot easier for another non-native animal to come over versus if you do it on an island, like you can feel fairly secure that you can protect that area over the long term. So they're really great refuges for seabirds in the past, and moving forward too. I think they're just going to become increasingly important.
And in the green room, we were kind of talking about how this is sort of just the beginning of your time as a grad student. Can you talk a little bit more about what your years upcoming are going to involve and what your sort of ultimate goal is?
Yeah, so I'm going to be looking at a computer a lot more than I used to be in the field. So I'm taking all of that data. So Channel Islands National Park, they have an inventory and monitoring program. But what I'm doing is actually I'm looking at all the data about seabird population trends over time, and I'm seeing how these threats are affecting the different kinds of seabirds and how they might affect them into the future. So then the park can use the limited resources that they have to try and help the seabirds that are in trouble. And I'm going to actually create a tool that the park can use to say, like, okay, if we only have X amount of money and we know that ashy storm petrels are in trouble, our best bet is to do artificial habitat, or whatever action that they can maybe do to help those seabirds. And yeah, so I'm just kind of in the beginning stages of starting to pull all that information together. So yeah.
Great. Well, thank you so much for presenting. I'm going to move on to questions for Anne. So let's see, Anne. This question comes from Sean, and he is wondering, does picking mushrooms hurt the ecosystem?
Yeah, so that's a really good question, and it's one that I've asked before, actually. So I guess this is sort of a cop-out answer, but it depends. So what are mushrooms? They're the fruiting bodies of the fungi, right? So their purpose is to act as the reproductive sort of bank of a fungi. So if a fungi wants to reproduce and spread to new environments or just make more of itself, it has to grow these mushrooms. The mushrooms then have spores in them, which are these tiny sort of seeds, seed-like structures, and they get dispersed in a variety of ways to colonize sort of new areas and to sort of increase the like that particular fungi. So obviously, if you pick mushrooms, you're sort of removing that capacity at that particular point of that particular fungi. That said, fungi produce a lot of mushrooms. And I think it's very hard to sort of pick so many mushrooms that you're going to totally cut off that fungi from being able to reproduce. So I think on like a single person scale or maybe even a group scale, it's probably unlikely that it has a severe detrimental effect on these fungi or like their health or longevity as a species. However, I guess if you like think about a massive scale and if you continually remove mushrooms for an area over a long period of time and really get all the mushrooms, yes, you could will eventually cause some damage to these species. But I asked about this from someone who grows these fungi and does a lot of work with them, and they said they actually did experiments where they picked a lot of these mushrooms from these experimental fungi plots they had, fungal plots, and it didn't harm the fungi at all. Like they just kept producing more mushrooms. So long answer, but it depends, and probably not.
Okay. Great. So this question comes from John. He's wondering, how can I improve the environment in my garden for fungi, compost, tilling techniques, etc.?
Yeah, that's a great question too. I'm an avid gardener, but I'm actually not an expert gardener, so I might not be the best person to answer that question. So yeah, I really don't have any specific advice. I mean, first of all, it depends on the kind of fungi you're talking about. Like I was saying in my talk, mycorrhizal fungi depend on plants to survive. They can't just live in the soil by themselves. So if you want to have those fungi, and those are the fungi that will help your plants survive as well, then you want to make sure your area is just covered with plants, because that's how these fungi will get established or continue to sort of exist. One thing I do is I actually just buy like mycorrhizal fungi fertilizer. You can get it in any garden store or a lot of garden stores, and literally it just adds more mycorrhizal fungi to your garden. You just scatter it around, and that sort of helps the mycorrhizal fungi establish and helps your plants establish as well. So hopefully that's helpful.
Yeah, for sure. I got a couple here from Ken, who's asking, and I'm going to say this word wrong, so help me out. How did they determine that a mycorrhizal network existed?
Yeah, you got it. Yeah, mycorrhizal.
Is that network disrupted by logging?
Yes, really great questions. Okay, so how did they establish the presence of a mycorrhizal network, right? Because like I talked about, like you can't see these things, right? They're all underground. There are these tiny microscopic structures. Well, so one way that a lot of researchers actually do physical experiments with these mycorrhizal networks or mycorrhizal fungi is they put in the ground, they put these things called mesh nets. And what the mesh nets do is, like, if you have a plant in the soil or in the lab, wherever, in a greenhouse, you surround that plant with this sort of giant mesh net all the way around on the bottom as well. And that allows for the mycorrhizal fungi that are partnering with the plant to go through the mesh net, but the plant roots cannot go through the mesh root because they're too big to fit through the mesh. And so if you establish these mesh nets, let's say around 2 plants, then the mycorrhizal fungi of both plants can grow through the mesh nets and connect in the middle, but the plant roots won't. And so then you can, let's say, there's a lot of different kinds of experiments, but people even label things like nutrients or carbon, and they sort of infuse it into one plant, and then this carbon is labeled so you can actually detect it with this special, like, instrument. Sorry, this is getting technical, but you put this labeled carbon into one plant, and if you measure it in the other plant, you know that it's gone through the mycorrhizal network and not through the plant roots. So yeah, it's sort of tricky, but yeah, it's sort of a tricky thing. But that's one of the ways that the establishment of these mycorrhizal networks was detected.
And then the second part of that is that network disrupted by logging?
Right.
It is.
Yeah, I mean, it definitely is. Like I was saying, mycorrhizal fungi depend on plant carbon to survive. So if you have a mycorrhizal fungi that's living with this plant, I mean, the relationship is so connected. They're almost like one organism, right? So if you destroy that plant, you cut it down, it stops photosynthesizing, it stops producing carbon. That carbon isn't going to go to that fungi, and that fungi will eventually not be able to survive. That said, these are mycorrhizal networks, and like I was saying, they partner with multiple plants and can extend for really long distances, especially some species of fungi can extend for meters and meters. Like one fungal individual can extend for a long ways. So, if you log like a certain area, but not do like clear cut logging, so like selective logging, it actually makes it easier for these fungi to survive because they can depend on the plants that weren't cut down for their carbon.
Great. And this is the last one for you, Anne. This comes from Olivia. She says, Anne, in the hidden life of trees, I learned that plants feel pain when preyed upon and send out defense signals. Upon further reading, I learned that not all researchers agree that this quote-unquote pain is like our pain. Could you comment on how plants feel pain and how that's communicated through underground networks?
That's a really intense question, but also really exciting because I think this is fascinating, right? This idea of like, are plants similar to us or are they not? Because in some ways they're similar, but they're also so vastly different. So I don't know a lot about that subject, but I do know that plants do evoke a kind of what they call a chemical scream when they're injured. So this isn't the same thing that we feel like when we're injured or like pricked by something, right? We have some kind of electrical signals that go up to our brain. They tell us, first of all, that we've been injured, and then they also send these, make us feel this pain, right? But you can also block those. But with plants, what happens is when you injure a plant and then you measure the defense chemicals in the air surrounding that plant and also within the plant, the chemicals change. So it's sort of like the plant is saying that it's injured, but in a chemical way, right? Because remember, plants use chemicals to communicate. So yes, the plant does respond to injury. Whether or not we can characterize that as pain is another question, but they do respond and they do send out chemicals to sort of signal that they're in trouble.
Okay, great. Thank you so much. I'm going to move on to Andrew and his questions. So let's see. The first one is from Jenna, and she says, is there an environmental cost to extracting Mars-like rock from the Mojave?
That is a very good question. I have not looked into that because it's a separate company that does the extraction. But NASA has kind of cleared this procedure. I should also specify that I have in total in all my 2 years of grad school, I have used maybe like one bag about that big of this sand. So it's not a whole bunch of it that gets made. That's interesting and something I should look into.
Okay. This next one is from Katie. What will happen to the surface of Mars if you extract the water? Will the dirt get looser or dustier?
Yes, on the small scale, it will. So if you extract the water, then you'll have a bunch of loose, dusty sand that will get kicked up really easily. So on Mars, you have these really intense dust storms because there's so much loose, really fine-grained sand that gets picked up very easily by the limited wind there. But we have to think about the scale here. So humans harvesting water from the dirt is only going to take a very small amount of water on the scope of an entire planet. So maybe if this process continued for hundreds of years, I don't know. I can't think that far ahead into the future. But on the small scale, absolutely, it'll dry out the dirt. But I don't foresee that being a big problem on the planetary scale.
Okay, this one is from Olivia. Andrew, could a Mars equivalent, and I'm going to say this wrong as well, could a Mars equivalent of tardigrades persist even in salty areas?
Yeah, so tardigrades, if you haven't heard of them, they're also known as water bears. They are these extremophiles, is what we call them. They're organisms that can survive extreme conditions that normal life could not. So for example, having intense radiation or intensely cold temperatures, or in this case, intensely really, really salty solutions. So for example, they've been found on the International Space Station, on the outside of the space station. And I think that they've been brought back inside and revived. They go into this hibernative state. So I think what Olivia's asking is if a similar organism exists on Mars, could it survive that? And that's a good question. I would pay so much money to know that answer. The thing is, we only understand life as we know it on Earth. If there is life on Mars, it could be very similar to our life, or it could be radically different. So I guess I should point out that on the scale that I showed during my talk of how salty is too salty, that limit includes tardigrades. So could there be a Martian microbe that's even more of an extremophile than tardigrades? Maybe. If so, we haven't found it yet because we've only sent up a handful of rovers really in our entire time. But who knows? I guess time will tell is my answer.
Okay. And this is from me, for the ultimate layperson who has not studied anything about the solar system since elementary school, why Mars? Why not, Venus? Why are we looking so closely at Mars?
That's a good one. The most immediate answer is that Mars is just the closest. It's the closest planet, so it's the easiest to get to. It's the cheapest. We can just launch a rocket there. But on a deeper level, it's also the most similar to what we have on Earth. And we think that in the distant past, one possibility is that Mars was very similar to Earth. Like I showed the pictures of possibly flowing water. We don't really see that on Venus. Venus is the next closest. Actually, you know what? Now that I think about it, depends on how you define close. But Venus and Mars are both our neighbors on either side. And the problem with Venus is that it has a intensely thick atmosphere that makes the surface temperature something like 900 degrees Fahrenheit. It's hot enough to literally melt metal. So we don't think there are great prospects for finding life there or establishing any kind of human exploration, but that's just where we are now. And so there are companies there are private companies and public organizations like NASA that want to go to Mars and have concrete plans laid out to send humans to Mars at some point within who knows how long, but soon.
Okay. And this last one is from Michael. He is volunteering to go with you to Mars and is anxious.
Excellent.
His series question is, will the Perseverance rover launching later this year be able to study the salty water on Mars that you've simulated in your lab?
Yeah, that's a good one. I actually included a little bit about the Perseverance rover in my talk, in my slides, and then about like 30 minutes before this, I was running through my slides. I'm like, no, it doesn't make sense, so I cut it out. But yeah, Perseverance rover, if you're not familiar, is also known as the Mars 2020 rover. It's going to be launched in July of this year. And starting next year, it'll land on Mars, actually in one of the deltas that I showed a picture of during my talk, looking at this ancient lake environment, trying to study and see if there are any signs of ancient life that might have lived in Mars's rivers and lakes, looking for chemical signatures or tiny fossils. But mostly it's going to be studying the rocks themselves, and not looking as much at whether water is present or whether life is present now.
Okay. Well, that brings us to the end of this program tonight. I want to thank you all again, Amelia, Anne, and Andrew, for presenting your sort of culmination of a year of work with us. And I wish you the best in your future schooling and research. It's all very exciting and super interesting. And thank you all so much for watching tonight. If you're interested in hearing more students' final sort of research presentations, the next one in our series is coming up next Thursday on May 7th. It will be at the same time, 6 p.m. Also, if you're interested in more town hall programming, you can click the follow button at the top of your screen for other events that we have coming up. So thank you all again so much, and have a great night.